Very good morning, students. In the present video lecture, I am trying to focus on the session 4, that is transpiration and structure of stomata, under first unit, that is plant water relations of paper 7, plant physiology and metabolism. Transpiration means water in plant body acts as a media to transport minerals, organic solutes, oxygen and hormones in the plant body. Plants generally absorb water either by imbibition or by diffusion or by osmosis. See, by any one of these methods, plants absorb considerable amount of water, quantity of water. And it is learned that only a small fraction of the absorbed water is utilized by the plant body for growth, development, and for various metabolic activities. Say, for example, 100% of water absorbed by the plant body. It is said that only 2 to 3% is being utilized for various metabolic activities, for growth, development, germination, etc. etc. The remaining 97% of the water is excess for the plant body. If that water is retained in the plant body, definitely the plant will undergo a process of decay. So nature has given the plants a mechanism to remove this excess water from the plant body. The rest of this water absorbed by the plants is ultimately lost to the atmosphere, either in the form of vapors or in the form of liquid droplets. Thus, we can define transpiration as the process of loss of excess of water from the aerial parts of the plant body, mostly through the stomata of the leaves, in the form of vapors, is referred as transpiration. So during transpiration, the excess water will be lost in the form of vapors, and most of the transpiration takes place through the stomata of the epidermis. Hence, such transpiration is also referred as foliar transpiration. Foliar transpiration. Transpiration occurs in almost all plants. However, in xerophytes, that means the plants which grows in desert conditions, xerophytes, the transpiration is tremendously reduced, very highly reduced. But the magnitude of transpiration remains fairly high in mesophytes. Depending upon the structure involved in the process of transpiration, transpiration is of three types. Number one is stomatal transpiration, number two is cuticular transpiration, and the last is lenticular transpiration. Stomatal transpiration means the loss of water which takes place through the stomata of the epidermis of the leaf and the stem when the stem is young is known as stomatal transpiration. Stomata are confined to the leaf and the stem. They are absent completely on the root system. The stomata in stem are present only when the stem is young. Through the stomata on the stem and also on the leaf, if the excess water is lost, such transpiration is called as stomatal transpiration. The magnitude of stomatal transpiration is about 95%. That means to say, the stomatal transpiration accounts for substantial loss of water from the plant in the form of vapors. That is, around 95% of the excess water is lost in the form of vapors through the stomata. Secondly, the cuticular transpiration. A small portion of the foliar transpiration takes place from the general surface of the leaf through an organ called cuticle. What is cuticle then? Cuticle is the waxy coat developed over the epidermis of the stem and the leaves. Why it is developed means cuticle is mainly developed to check, to control, to avoid excess transpiration. However, due to cracks which are developed on the surface of the cuticle, certain amount of internal water gets vaporized. Thus, the transpiration which takes place through the cuticle of the epidermis of the stem and the leaves is known as cuticular transpiration. As told earlier, the magnitude of cuticular transpiration is less when compared to the stomatal transpiration, and the magnitude of cuticular transpiration amounts to about 4 to 10% of the total amount of transpiration. Lastly, the lenticular transpiration. What are lenticels? Lenticels are the small aerating structures or pores developed on the bark of certain woody dicot plants. Usually, these lenticels are developed after secondary growth. Even through this type of lenticels, transpiration takes place. Such a transpiration which takes place through the lenticels present in the bark of the old dicot stem and fruits is known as lenticular transpiration. The magnitude of lenticular transpiration is almost negligible, where it amounts to about 0.1 to 1% of the total amount of transpiration. Here, coming to the structure of stomata, the second part of the discussion of this video lecture. Stomata are the minute microscopic openings present on the epidermis of the leaves and the young stem. They cannot be seen through your eyes. They are microscopic in their nature. They are confined to the epidermis of the leaf and also the epidermis of the young stem. In old stems, stomata are replaced by lenticels after secondary growth in some plants. The number of stomata, distribution of stomata, and the frequency of the stomata varies from species to species. It is learned that number of stomata range from 5,000 to 30,000 per square centimeter. It is an average number. 5,000 to 30,000 per square centimeter of stomata are found per square centimeter of the leaf area or the epidermal area. This stomatal apparatus, this is the diagram of the stomatal apparatus. Stoma, nothing but pore. Aperture. The entire structure is called as stomatal apparatus. Stomatal apparatus consists of, as you can see, an elliptical aperture bounded by two modified epidermal cells. The cells which are present around the elliptical cells are also epidermal cells, but they are modified in such a way that they surrounds these uh, stomatal cells. Here they are the elliptical aperture bounded by two modified epidermal cells called epidermal cells. Since these epidermal cells guard the stomatal opening or the pore, they are referred as guard cells. They are referred as guard cells. These guard cells are photosynthetic in nature, in function. The guard cells are in monocot plants, they are dumbbell in shape, and in dicot plants, they are kidney shaped. As you can see in the diagram, they are they are bean shaped or kidney shaped, and it clearly says that it is the stomatal structure of a dicot plant. Look at the wall of the guard cells. It is unevenly thickened. The wall of the guard cell is very much thickened towards the stomatal aperture, and it is thin in the rest of the sides. The wall of the aperture, wall of the guard cell towards the aperture is thick and inelastic, and it is thin and elastic in rest of the sides. Each guard cell contains a nucleus, vacuole, cytoplasm, and number of chloroplasts in the cytoplasm. So we can say each guard cell contains vacuolated protoplasm. Vacuolated protoplasm. The cytoplasm contains number of chloroplasts. The guard cells are usually much smaller in size when compared to the epidermal cells. The guard cells are surrounded by two or more epidermal cells called subsidiary cells. Otherwise, they are also called as accessory cells. Accessory cells. Some of the qualifying features of guard cells are special features of guard cells, means to say they are the shape of the guard cell itself is a special feature. The dumbbell shape in case of monocots, and the bean shaped or kidney shaped in case of dicots. Unevenly thickened cell wall is again another feature, special feature of the guard cells. And third is presence of number of chloroplasts in the cytoplasm. Fourth is inelastic, unevenly thick ventral wall and flexible dorsal, thin dorsal wall. Flexible, thin dorsal wall. And the starch is accumulated in these guard cells. Generally, this is the structure of the stomata or stomatal apparatus. This kind of stomata are generally open in daytime, that is during daytime when there is a sunlight, and closed during night time. When these stomata are kept open during daytime, transpiration takes place through the stomata in the form of vapors. Along with exchange of gases. The stomatal openings are mainly to facilitate the exchange of gases. The main function of stomata is to facilitate the exchange of gases. But when these stomata are kept open for the exchange of gases, the excess water is also lost in the form of vapors. That is why these plants have to bear the evil of transpiration in order to get the benefit of exchange of gases. That is why Curtis, scientist, considered transpiration as necessary evil. Necessary evil. So, in nutshell, the transpiration is loss of excess of water in the form of vapors, which takes place through the foliar regions of the plant body. The organs through which transpiration takes place are: number one is stomata of the epidermis, number two is cuticle on the epidermis, and number three is lenticels. Accordingly, the transpirations of three types, that is stomatal transpiration, cuticular transpiration, and lenticular transpiration. As far as the structure of stomata is concerned, stomata is a minute microscopic opening which is present on the epidermis of the leaf and the stem when the stem is young. It cannot be visualized through our eyes. This kind of minute microscopic opening is guarded by two special modified cells which are called as guard cells. These guard cells are dumbbell shaped in monocots and kidney shaped in dicots. The wall of the guard cells is unevenly thickened. It is thick and inelastic towards the aperture and thin towards the rest of the sites. Each guard cell contains vacuolated protoplasm with a number of chloroplasts. Guard cells are surrounded by subsidiary cells and epidermal cells. This kind of stomatal apparatus is open during daytime in presence of light and closed during night. Such plants are called photoactive plants. When stomata are open during daytime, transpiration takes place through the stomata. Thank you.